Welcome to Oster Oncology. This is Dr. David Oster, and today's talk we will be focusing on survival skills of a cancer cell and how these survival skills are actually targets for our treatments. So in this video, I'm going to review the pivotal survival skills that allow a cancer cell to grow, to spread, and even at times to resist treatments. And all of these survival skills can also be referred to as drivers, so you may hear me speak of them as drivers throughout this talk. I've reviewed them here by location, so you're going to see me present them this way by location. So some of them are associated with the cell membrane. Some of them will be presented as external triggers to cellular growth, some internal, and so forth. So you're going to see me present them this way. And this overview is not presented just to geek out over or bore you with the cellular details and machinery of cancer cell growth, but rather I want you to gain an appreciation of where and how conventional treatments have been designed to have their impact. They've been ingeniously developed to shut down some of these drivers of disease, but I also want you to see that conventional treatments do not have answers for all of these abnormal acquired growth advantages. So many of these drivers are left unchecked and allowed to roam free within a cancer cell. And in light of that, I want to show you at the end of this talk, so please stick around to the very end, I'm going to show you where the conventional treatments can plug in to shut down some of these drivers, but many of these survival skills have been shown to be sabotaged by natural compounds. So I really want to show you how these natural compounds, when used safely and with someone that has experience and knowledge in this, can be used in conjunction with conventional treatments to get a more comprehensive shutdown of some of these drivers. And we do know as well that cancer cells have not just one driver active, but usually a good handful of many of these drivers are together at the same time, concurrently active in a case. So stay to the end and I'm going to show you that. So you'll see with the illustration already, I've started with the membrane-associated survival skills. So receptors is one that many of us can be familiar with already. So for instance, estrogen receptors in a breast cancer case, these are receptors. So you see them drawn here in green, being triggered from the outside by those blue little circles. So those could be a hormone or a growth factor, and when they are triggered, they send an internal signal inside the cell for the cell to be healthy and for it to grow. This is very normal in all cells, but you'll see how this can become exaggerated in cancer cells. The next one that I'm showing you here associated with the membrane are these little protein structures, and they are pumps. They are efflux pumps. So as opposed to influx, bringing something inside, these are efflux pumps. They effectively pump toxins and chemicals and compounds from within inside the cell environment and spit them out. And so these efflux pumps, in particular to the discussion today as an advantage to a cancer cell, we are referring to these because they can very effectively kick chemotherapy, which we really want concentrated with inside a cancer cell. They can effectively kick them out. And so there's a number of these multi-drug resistance proteins. So MDRP1 is a class of multi-drug resistance protein pumps. These PGP and ABCB1, these are just other classes of efflux pumps. So if I zoom in here and magnify this for you, I'm going to first draw, here's the membrane. So we often draw a membrane this way up close with these circles and lines, and then embedded within that membrane, you see these pumps. And I'll draw it like a barrel here, but again, you see that it spans the membrane, so it has access both to the inside environment of the cell, which would be on the right, and then on the left is the outside environment. So it's embedded within the membrane. And I'll draw some chemotherapy molecules here in the blue. And again, we want that chemotherapy concentrated. So you'll see I'll put some square brackets around the chemo here. And when we're looking at illustrations and diagrams in biology, whenever you see that square bracket, it refers to concentration. So we want chemotherapy concentrated within the cell. But again, these efflux pumps can very effectively spit the chemotherapy outside, and we are not getting the desired impact that we'd like to see from the chemotherapy. So you'll see that the chemotherapy concentration is effectively reduced there. So right next to that, here's the lipid bilayer again, the outer membrane of the cell, and I'll zoom in here on a membrane and the receptor that's embedded there as well. So I'll draw a little receptor here, and again, receptors span the membrane. It has direct communication with the outside, and so here I'm drawing a growth factor. It could be a hormone, it could be estrogen, it could be another growth factor that docks into the receptor. And these receptors are designed specifically for one type of trigger. And when they bind, the internal aspect of the receptor then communicates that a binding has occurred, and it sends a signal into the cell to grow and be healthy and maybe to divide even. And this is normal. This is the normal result, the expected designed result of that receptor. The problem arises, though, and again as an advantage, is when this normal function is hijacked by a cancer cell, and either the distribution and the number of receptors is increased on the membrane, so it just covers, the cancer cell covers itself with receptors, being far more welcoming to many more signals from the outside environment, or their function has been really jacked up, and it effectively at the end here, as with all these arrows, it's not just telling a cell in a normal fashion to grow and divide, but to grow, divide. And so I'll put a little exclamation mark here just to show that this is exaggerated, and then of course becoming dangerous for the cell to be growing at a far more rapid pace than we want it to. And so I'll draw a little red arrow here. It is the end result. The end point is really getting to the nucleus, and we'll get to that in a little bit further in the talk. But again, this is one area and location where we can have advantages acquired by a cancer cell, and this really is at the cell membrane, these factors that can play a role. So the next area that I want to focus on is what happens immediately after these receptors are triggered. And so I'm going to be drawing some receptors here again. We're not again going over the membrane associated survival skills, but it's what happens in the aftermath of the receptor. So what we're talking about here is actually the internal signaling from a receptor to the nucleus and the DNA. So what I'm drawing here are these molecules from the blue to the green here. You'll see that there is a cascade of which means inside, intracellular molecules that trigger a domino effect, a cascade effect of activation of one molecule to another, and ultimately telling the cell to grow and maybe to divide. And again, this is normal. But the problem arises when this becomes hijacked and exaggerated by a cancer cell. So again, I'll illustrate this and magnify it a bit here, because a lot of the details, I'm going to name some of these molecules quickly for you, because a lot of these molecules, even if one of them is mutated, one of these domino effect molecules is mutated, then we can get a very exaggerated growth signal inside the cell. So you'll also see a lot of the conventional treatments can target many of these specific intracellular molecules. This is targeted therapy. And so here's the membrane again, receptor X. And again, this title is intracellular signaling, so we're not necessarily speaking of any specific receptor, but what happens after they get stimulated. So on the internal aspect of these receptors, you're going to see me draw some little blue circles. And these are the immediate signaling molecules that first communicate that a receptor has been turned on from the outside. And so they go by name. There's many different cascades. This is RAS, and then I'll draw STAT3, STAT3, and PI3K is a very common mutation in cancer cells, PI3K. Now again, they communicate after they're turned on, they turn on and activate other signaling molecules. And so you'll see that RAS turns on a molecule called RAF. STAT3 works with JAK. You'll see me label them here in a second. and then PI3K works downstream as well to activate ACT and mTOR. So here's RAF being activated from RAS, there's JAK, here's ACT down to mTOR, and then MEK, MEK, and MAP kinase, MAPK, and MEK actually activates another molecule called ERK, ERK. So when we get to the level of mTOR, ERK, JAK, MAP kinase, it's at that level that these are actually molecules that communicate directly to genes within the nucleus on the DNA and stimulate growth and division in a cell. Usually they are under a very close watch. A very close braking system is monitoring these so this doesn't get out of control. But if we have mutations in any of these, we can have these cascades greatly exaggerated. One of the commonly known important braking systems on the PI3K pathway is a molecule called PTEN. It's a beautiful tumor suppressor gene product, and it shuts down, I'll put a little minus symbol here. It is very important that when PI3K activates AKT, ACT, that it only does it once. It does it once and then PTEN shuts it off. But again, if we have a mutation in PTEN and there is no brake, all we have is full gas on these transcription, on these pathways that are internal signaling. So the third area that we're going to move to quickly here is the external environment, and there are a number of factors here that I'm going to highlight for you quickly, but these are external environmental factors. They can be in the circulation that we can have an impact on. They can actually be encouraged and fostered by the cancer cell itself. So this will be the third category that we'll look at. I'll number them here just so we stay on top of this. So the first one is the cellular membrane factors, and let me just do this for us here, first and second, and here's the third one. So let's discuss these external environmental triggers and factors that cancer cells harness and really rely upon for their survival and growth. So the first one that we can discuss here is hormones. So circulating hormones and growth factors, these triggers that stimulate receptors, which we initially spoke of at the start of the talk. So these hormonal triggers bind onto receptors. So there's a number of them here. They can be hormones like, for instance, estrogen, I'll put here with an E, or they can be testosterone, DHT, which is dihydroxytestosterone, which is a potent form of testosterone. They can also be growth factors, epidermal growth factor, EGF, vascular endothelial growth factor, VEGF, and then insulin-like growth factor 1 is a common one, very common as well, that stimulates growth. So they are external. Cancer cells love them, thrive off them, and their impact is mediated through receptors. Number two, we can look at in a very same manner from the outside, we can have inflammatory compounds trigger cellular growth from the outside. So these inflammatory compounds, probably less commonly known to you, but some of them are interleukins like IL-6, prostaglandins like prostaglandin E2, and then tumor necrosis factor alpha. There are many others though, but those are the common ones. Certainly interleukin 6 is a big one, and again from the outside stimulating growth to that cancer cell, very much appreciated by that cancer cell. Inflammation feeding cancer. A huge impact you can have on inflammation is your diet actually, but we can talk about that later. Internally, inflammation is communicated to the cell by NF-kappa B. It is a major factor. I may do a talk just on NF-kappa B in a later illustration, but NF-kappa B is like wind in the sails of so many other survival advantages and skills. So for instance, NF-kappa B can put wind in the sails of genetic changes inside a cell for growth. It can enhance what's happening at the receptors. It can really crank up an already abnormal signaling cascade internally, like we've already illustrated. And so NF-kappa B is a major inflammatory mediator of rapid abnormal cellular growth and spread of cancer cells. The third piece here is quite unique in what it does. These are matrix metalloproteinases. And matrix, we'll call them MMP for short here. And these MMPs are very unique. So they are numbered, and there's different ones that certainly influence the immediate outside environment to a tumor. And again, these are numbered. The important ones are matrix metalloproteinases 2, 3, 6, and 9. And they are actually released by a cancer cell. So cancer cells will crank these out and put them out into their immediate environment all by themselves. And it's just interesting, again, NF-kappa B can really crank up the release from a cancer cell and from a tumor of these matrix metalloproteinases. So what do they do? What's so important? It's very unique in what they do. So immediately outside of any type of cell in the body, but certainly of tumors as well when they start to grow, there is connective tissue that holds a cancer cell in place. It holds it there so that a liver cell stays in the liver, that a breast cell stays in the breast. They don't move around. They are kept in place by connective tissue. And so what a cancer cell can do is release these MMPs, and I'll draw a little set of scissors here. They are molecular scissors. So matrix metalloproteinases, when released into the immediate environment, can start to cut away at the network of this connective tissue that provides structure. And what happens is when it becomes loosened, a cancer cell then takes the advantage of moving through that connective tissue and therefore can be the launching pad of cellular spread out of its original site. Metastasis is what we're referring to here. So matrix metalloproteinases are important for that. The fourth piece here of the external environmental factors that I wanted to discuss really is based on the vascular endothelial growth factor, which I just sort of pointed to there, the VEGF in the hormonal triggers bracket there. And it is very important for blood supply to a tumor. So if I draw some blood flow here, some capillaries flowing normally through a tissue, what a cancer cell can do, and we also have this in circulation as well. It can be released by cancer cells, but we can also have this just generally high in the circulation, this vascular endothelial growth factor. And when it is circulating, the nearby blood vessels to a tumor, so I'll draw the VEGF molecules here, when they are locally released in high concentration, the blood passing by the blood vessel will actually branch off and bud, a budding of new blood vessels in the direction of where that vascular endothelial growth factor is being released from. So you see that higher concentration and then a budding, and then we get an enhanced blood flow, bringing more inflammatory factors, bringing more hormonal factors, bringing more nutrition and building block resources to the tumor for growth. So this is called angiogenesis, genesis meaning creation, and the creation of new blood vessels to the local environment. So these are some of the external survival skills that cancer cells acquire and rely on for their growth. Another unique external one, but it's sort of unique, so I'll do it on its own here, and it also has a very unique new therapy, immunotherapy. So if we talk about the immune system now, and the immune system is made up of your immune cells, so the cells that keep you safe from bugs and viruses and are involved in healing of tissue that's damaged and just keeping your body sound from infections and illness. The immune system here, I'll draw them here. So you've got a host of immune cells, and they are always aware of any type of malignant cells in the body. And so they are always on the hunt, and when they find a cancer cell, they will engage a cancer cell. So if I magnify it a little bit here, and this is not to scale, it's not that the cancer cell dwarfs an immune cell so greatly here, but just for scaling purposes so I can fit everything in, I'll just draw a slightly larger immune cell here. And this immune cell will engage a cancer cell, and it does it through receptors. So that immune cell would love to gobble up and dismantle this cancer cell. But when it comes close, cancer cells can then project a receptor, a membrane-bound receptor, and when it binds onto the immune cell receptor, which I'll label here, the immune cell receptor is called PD-1. And the cancer cell will present to that immune cell when it comes close another receptor called PD-L1. And PD-L1 will effectively shut off, it's almost like a hypnotist, just putting to sleep that immune cell. So that immune cell approaches all ramped up, ready to engage that cancer cell, but the cancer cell itself says, come on, come close, and I'm going to shut you off, and that's effectively what they do. So we call this immune evasion. So your immune system is a cancer killer, but now we're seeing more in the research, definitely we're getting a greater understanding that almost all cancer types and cancer cell types rely on this immune evasion so that they can resist treatment, resist the immune system. Okay, so we're moving into the nucleus now. So another compartment, we've been on the membrane, we've been inside the cell with intracellular signaling on the outside, and now we're moving in to the actual nucleus. And inside the nucleus of a cell is where the DNA is stored. You see the green chromosomes there with our DNA tightly wound and compartmentalized in there. And then along that DNA we have genes. So if we magnify here into this realm, the fifth point here is what's happening at the genetic level. And we know that cancer, the primary dominant understood cancer theory, is that we have mutations. All of these other survival skills are fueled from mutations at the level of the DNA. So genes have become mutated. And it really comes in two different forms of mutations. There's two classes of mutations, and they're sort of mirror images of each other. There are oncogenes that can be mutated. And oncogenes are, and this is of course bad, oncogenes are some of the advantages that already tell a cell to grow, but when they get mutated, it's an enhanced signal to grow even more. So for instance, some of these we've already discussed: PI3K, STAT3, RAS is another, MEK is another. So, and then again, we saw these up at the internal signaling cascade. So when they get mutated, they can become deaf to the brakes that are usually controlling them. When they get mutated, they become deaf and they no longer hear the stop signal, the control signal, which are usually, the stop signals and the brakes are usually from genes that we call tumor suppressor genes. These are good, but they are the very essential brakes on cellular growth. When they become mutated, when those genes that are responsible for producing, for instance, P10, which was the brake for PI3K, when that becomes mutated, it loses its function. There's another very important tumor suppressor gene product called P53. We'll be discussing that in just a second as well, but it's the loss of their function that can encourage cellular growth. Now, we can't do anything at this point in time to a mutated gene. So when a gene becomes mutated, we can't correct it. There's no supplement, there's no chemotherapy or drug at this point that can correct an oncogene mutation or a tumor suppressor gene mutation. We can't do anything there. But we can have impact when there are advantages gained at the genetic level, not from the mutation necessarily, but from a density of the coiling of the DNA. So I'm going to explain this to you here. This is not, so I put in the box there, not mutated, hidden, silenced. These genes I'm referring to can be hidden or silenced, and it all comes down to the density of the coiling of the DNA. So we can have impact here. This gives us some hope, even at the level, even in the scenario where a gene's already been mutated, it gives us a lot of hope here. So I'm going to draw some DNA here. You'll see the double helix, and I just showed you that it's sort of very tightly squeezed together. Picture an old school telephone cord. So when I was growing up in the house, my folks had that old phone with a long telephone cord that reached across the house. And when it was connected to the base very close, the coiling was very tight. And so if a gene is hidden within this tight coiling, I'll put a little volume signal there, it's effectively off. It's not really impacting the cell. But when we see that same gene, that same segment of DNA stretched out wider it's open to having an impact and an influence. We often say it's being read. It's being read and listened to. It has a higher volume, and it is effectively on. That gene can be turned on and off by the coiling of the DNA. So what we see in cancer cells is that tumor suppressor genes, whether they're mutated or not, we see generally this pattern that tumor suppressor genes in cancer cells are coiled up tight and are off, and oncogenes are spread open and are on. And this is a general pattern you see whether the PI3K is mutated or not. So there is a preference to having growth speaking louder than control based on the coiling of the DNA. Okay, so the last section here, thanks for staying with us because we're going through a lot of detail here. I'm going to draw another little beautiful little organelle. It's a little factory inside a cell. And there are so many of them. I'll draw some smaller versions of it right close by here. There are hundreds and thousands within a single cell of these little tiny, we call them organelles, small little organs within a cell. And this one is called the mitochondria. Some of you that have done cell biology may be familiar with mitochondria. And mitochondria, under normal circumstances, just a cell, clicking along, it's responsible for energy production in a cell. So it's the energy factory within a cell. But it also plays an immense role in cancer. So it plays an immense role in cancer when we get to the point that dysfunction is clearly seen within a cell. So there's a word here called apoptosis. So apoptosis is an ability that a cell should have, we want it to have. Cancer cells shut this ability off. It's an important cell to protect us from cancer, apoptosis. And apoptosis is a very important function that happens at the time of error. When I say error, I mean errors in cellular growth. All of these other survival skills we've already discussed. When it is clear that this is happening in a cell, apoptosis ideally should kick in and it should stop cell division. We do not want to pass on this version of a cell to daughter cells. So the cell should, if apoptosis is active, stop a cell dividing and then call a timeout and proofread the DNA, look for the mutations, and find the error. When that error is found, it's an amazing advantage, ability that all cells have to stop cell division and proofread itself, double check itself. It's the conscience of the cell, really. And there's two outcomes that can happen once that error is found. If apoptosis is functioning, it can stimulate repair of that. So there actually is repairabilities of mutations of all cells. But if we cannot repair it, then the cell will enter into cell death. It'll actually dismantle itself. It's cell suicide. So cell suicide is an essential function to all cells. So I'm going to show you how this plays out. I'll just magnify a little bit here what a mitochondria looks like, because I want to highlight for you some of the key players here. And if we lose the presence and function of these key players, we lose the ability to stop cell division. We need this. We lose the ability to proofread. We need this. We lose the ability, most importantly, to kill off a cell that's abnormally dividing. So generally we know that apoptosis is, and I'm going to look at P53 here. P53 is really important. It's a tumor suppressor gene product. And it also plays off of something called BAX and BCL2. So these are really important for the mechanism. So I'm going to show you, I'm going to walk you right through here how a cell kills itself, how it triggers dismantling of itself. So I'll draw BCL2 here as a little black dot and BAX as more of a rectangular blue. And they find themselves right in the outer membrane of the mitochondria. So just like a cell has a membrane, so does the mitochondria. And so BCL2 is found there, and it's usually bound to BAX. And it inactivates BAX. It actually keeps BAX under lock and key. BCL2 inhibits BAX. Now, if BAX is activated and allowed to act the way it's been designed to, it forms a pore. So a whole bunch of these receptors get together, and I'll draw sort of what the mitochondrial membrane, it'll find itself forming a pore. in the membrane. Now, this is prevented by BCL2 when it's bound to Bax. But if Bax is released on its own, or if Bax expression, if its presence is increased in number, it'll form that pore, which is important because I'm drawing here in the center of the mitochondria something called cytochrome C. Cytochrome C is usually quarantined inside the mitochondria. But when Bax forms that pore that it's designed to do under times when a cell has decided it should die, it will form that pore, and that pore is designed to release cytochrome C from the inside of a mitochondria into the greater compartment of the cell. So now it's free in the cell. The cytochrome C is outside the mitochondria here. And the significance of that is it triggers the cascade for cell death once it's released. So I'm drawing here this sort of star-shaped, it's just an amazing molecule called an apoptosome. And of course you'll see the root word, we're talking about apoptosis and cell death. This is an apoptosome. And apoptosomes can bind, it's sort of a 7-starred structure. It can bind 7 cytochromes on its tips, cytochrome C. So a cell will never experience this unless it's decided that it needs to die. Cytochrome C usually is quarantined, but when it is released, the apoptosome is engaged by cytochrome C. When it does that, it becomes this amazing springboard to activate these final molecules of cell destruction. So I'll show you the black squares are inactive, but the apoptosome, I'll have them swing in here to become activated. And I'll draw these little Pac-Man here. And it's an operative little symbol because they really do chop up cell structures and eat up the architecture of a cell that holds a cell together and just dismantles the cell. These are called caspases. So again, caspases are inactive when a cell is doing great, but when a cell is decided to enter into apoptosis, caspases are the effective executors of the cell, and they're numbered as well. In numbers, caspase 3, 7, and 9. And once they are released and let loose in a cell, it's lights out for the cell. So again, what we're looking at here is a dismantling of the cell. So the inhibition, the silencing of this whole cascade, apoptosis, cancer cells shut down apoptosis. They do not want to hear this. They do not want to have to deal with this because it is cell death to them. So a cancer cell, and it's not this, I'm sort of talking as if a cancer cell has motives, but molecularly, apoptosis is shut off either by a mutated p53 or a decrease in the Bax and other mechanisms that can really hinder apoptosis. And so even if a cell is getting hit by chemotherapy, if this is shut off, you're not really going to get the full dismantling and killing off of a cell, regardless of how toxic or under pressure a cell is. So I'm going to give you a little, this may be geeking out a little bit here, forgive me, but P53, I'm going to put 2 boxes beside P53 because it does have two different genes. So we do have 2 copies of every gene. So often you only have mutation, I'll draw a little X here, as the mutated form, but you usually have a functioning P53 gene. The problem is P53, when it works, when it effectively activates this cascade, it does it in a group of 4 molecules. So each gene makes 1. And so it also affects the gene. So to show you here, the P53 can affect genes in the nucleus as well. But when it also impacts the mitochondria for apoptosis, even if one of the 4 subunits, even if we have 3 of the 4 that are great, it will not work. So if there's one mutated form of the 4 subunits that are needed for effectiveness, then we do not get P53 active. So we have both of these genes playing off each other. And effectively, usually in cancer cells, the mutated form is highly active. But again, we need 4 subunits forming a tetramer. Tetramer means 4 together. And that is an effective form to initiate the release of cytochrome C through that Bax pore. So that's not happening. We're not getting apoptosis. Okay, so that's everything. Reviewed it in these different areas, these different locations where cellular advantages can take place. What I'm going to do now is walk you through where I'm going to do a little purple dot here and a little orange dot to show you where conventional treatments can be active. One thing to keep in mind that even though conventional treatments can act in all these different areas, because of toxicity, we can never deliver them all at once. So for instance, receptors, if you notice the receptors there, I'm going to put in a little purple dot because we do have things like tamoxifen. The orange dot shows you where we know we have research showing that natural compounds can be effective in those areas too. So let's just go, I'm going to do this really quickly here. So intracellular signaling, we know, I've got the conventional purple pen here. So this is chemotherapy or targeted treatments. We know that we have inhibitors for RAS. Well, let me just show you here. So generally we can hit all of these major targets. So we have mTOR inhibitors, we have MEK inhibitors that I just highlighted there for you. In the external environment, we know that we have aromatase inhibitors that can decrease estrogen levels. We can decrease testosterone levels. We have EGF inhibitors and VEGF inhibitors. So conventional treatment can be effective there. Inflammatory triggers, matrix metalloproteinases. There are no conventional drugs, so I'm not going to put a drug beside the inflammatory chemicals. If we look at immune evasion, yes, we do. We have immunotherapies now, PD-L1 and PD-1 inhibitors, and it's been a major advancement in conventional treatment. If we look at mutations, there's no known drugs that we know that can change any of the unraveling. And when it comes to apoptosis, there's no drugs that can help that either. So those are left in conventional treatment as untouched wildcards. If we look at natural compounds, the efflux pumps, again, we have no drugs to impact the efflux pumps, but we do have natural compounds. With the intracellular signaling, across the board, we see all of these cascades can be impacted by things like green tea extract, turmeric, resveratrol, indole 3-carbinol, the list goes on and on. P10 can be activated as well, which conventional treatments do not impact. Again, we have impact here with the natural compounds, especially the inflammatory compounds and NF-kappa B. There's no drug that impacts NF-kappa B. Some of the biggest ones for that would be reishi mushroom, curcumin, and such. Matrix metalloproteinases can be impacted as, again, curcumin and others show that they can decrease circulating MMPs. Looking at the level of mutations, we can't do anything to correct a mutation that's already there. We can't do anything. But there are compounds that can increase the expression and reverse, so we can make the oncogenes the quiet ones and the tumor suppressor genes the loud genes in a cell. So for instance, we can increase P53, the actual functioning gene, and increase the likelihood that we're going to be making a pure tetramer of P53, activating BAX and allowing apoptosis. That whole mechanism, caspases to be released, all of this has been shown in studies. We can activate that, reclaiming the ability to activate apoptosis in a cancer cell. So one last piece too with the immune system, another big piece, even though we can inhibit the cancer cell shutting off of the immune system, but another angle to enhance the immune system's ability to kill off cancer cells is to jazz up immune system function. So we use a lot of herbs and mushrooms for that as well, as long as they're done in safe conjunction with treatments. So again, I just wanted to walk you through this overview of what we see. The conventional treatments are designed in very ingenious, specific ways, but they leave so many wild cards as well. And so I wanted to walk you through and illustrate how we can see natural compounds fill in the gaps in cancer care.